Well, hey, AP Biology, hope you're doing well when you get this video. So the goal of this video is to help you understand exactly what I'm looking for in the Catalase Lab report. And so that's the lab that we did a few weeks ago, a week or so ago. So here's Catalase again. We've been learning about structures as far as protein structures goes, primary, secondary, tertiary, and quaternary. And so primary, of course, is amino acids. Secondary involves hydrogen bonding, alpha helices, beta sheets. Tertiary involves all the other bonding due to R group interactions. Remember R group interactions? Very important to think about that. And so these are multiple different tertiary units combined, subunits combined together to form a functioning protein in a quaternary unit. So catalase, again, breaks down this reaction right here. So it takes this, the substrate, hydrogen peroxide, and it breaks down into the products of water and oxygen gas. And so the oxygen gas is literally why we were able to really do this experiment. Oxygen gas directly related to enzyme activity. You know, the more oxygen gas is produced, we can infer different things about enzyme activity. So hydrogen peroxide, again, is a dangerous substance. It's from metabolism, so we've got to convert into other substances that aren't dangerous for the body. So catalase is frequently used by cells to rapidly catalyze, again, this is a catalyst, the decomposition of hydrogen peroxide. So we were investigating catalase in this experiment and what exactly affects its activity level. So here's a great, kind of diagram of enzyme substrate complex. You can see here that here's hydrogen peroxide, which would be the substrate. Each enzyme has an active site. Remember what we talked about? Form leads to function. Okay, and so here is the enzyme substrate complex induced fit model right here, induced fit. And then you have the reaction taking place and releasing water and oxygen into it. So we can adjust the rate of enzymes based upon their environment. So this was, student, teacher-directed inquiry, but you obviously designed your own experiment. And so I had these options for you in lab. You could test pH, you could test temperature, salt, and concentrations. And I believe everyone in this lab either chose pH or salt, okay? And so that are great choices. Again, not really a wrong choice, but it's interesting definitely what we saw in data. So here is the general experimental setup. Remember, we were doing the floating disk assay, so we basically let it float to the bottom, started the timer, and then when it hit the water, and then when it flowed back up, we stopped the timer. So it hits the water start time, hits the top stop time, and then we measured that, and that's directly what we started thinking about, directly related to enzyme activity. Okay, how active the enzyme was, has it been denatured, what's happening, because if catalase is on the filter paper disc right here, it's going to be metabolizing, forming those, that oxygen product, allowing bubbles to form within the filter paper and therefore rising to the top. And so this is just a general experiment. I will ask you to draw this out. So here's your lab report. So this is exactly what I'm looking for. We've already discussed this in class a little bit, but just wanted to make a video to try to help you understand more specifics. But so again, introduction should include just some basic understanding of what's happening with enzymes, like how do they work, why is catalase important, basically I already told you all that. And then the purpose, yeah, I mean, again, in this course, why are we doing this? What are we learning about? Hypothesis is an if-then statement. So I do want you to draw your experiment, the test tubes, just kind of label it, draw and label it. Labeling would be good here, so I understand. I should be able to look at your picture and understand exactly kind of what you did. A table showing your data, and then a correct graph, okay? A correct visual representation of your data. And I'm not really going to give any hints with that. I do want you to think about the best way to, again, tell a story using your data. And then finally, the lab report itself. Conclusion. So here's your conclusion. Everybody's conclusion should be different. So was your hypothesis supported or rejected? And why? So again, we're using data here. We're not just like guessing, we're using data. From your data, explain how your variable affected enzymes activity. So yes, might want to do a little research here, but I will tell you right now, it probably is due to bonding. Remember, it's all about form, right? If form changes, then the function changes. Something's happening if form leads to function. So if an enzyme's form changes, the function, it might be slower, it might not even work. So think about that. Was your enzyme denatured? And how do you know? Again, you should use data with that. And then how does this lab relate to the statement form leads to function? We've kind of been talking about it for a while, but this is a very important topic in biology and the foundation for really the central dogma and also many different disorders and diseases. And as always, I want you to think about possible ways that we could improve the experiment by canceling out errors. Okay, so hopefully this video was helpful to you. Hopefully when you watch it, you're doing well. So excited about the rest of this year and what it entails. If you have any questions, please let me know. God bless.